Cancers are genetically heterogeneous. The same anti-cancer drug may have varying degrees of effectiveness on patients due to their different genetic profiles. Oftentimes, it is a trial and error process, and patients have to try many different anti-cancer drugs that are ineffective with significant side effects before finding one that is effective. Cancer cellular data are extremely large and complex and difficult to process, and with the development of deep learning, it has been applied to many bioinformatics fields with great success, such as investigating DNA variants and extraction of genomic features for survival prediction. The goal of this research project is to use deep learning methods to analyze large amounts of complex biological data that is challenging to process with conventional analytical methods, and to predict the responses of tumor cells to different anti-cancer drugs using a multi-omics approach, so that doctors will be able to take a more customized approach to prescribing anti-cancer drugs that are likely to be more effective, and to also experiment with different methodologies to derive better prediction of drug responses. Why do we need a multi-omics approach? Multi-omics techniques use multiple types of omics data, such as genome, proteome, transcriptome, and metabolome, and so on. They provide a more holistic molecular perspective of biological systems. Cancer mechanisms are highly complex, and it is difficult to achieve accurate prediction based on a single facet, such as genomics solely. Therefore, we'll be using multiple omics of tumor cells for prediction of drug responses. The multi-omics data that we used in this research project are gene expression, metabolomics, RPPA, gene mutation, and gene copy number variation. We will be predicting the drug responses of tumor cells, and the drug responses are measured by IC50, which measures the effectiveness of a substance in a biological environment. The lower the IC50 value, the more effective a drug is, and the responses to the top 20 most commonly used anti-cancer drugs are predicted. In this project, we created one DNN for each individual omics, and we created a DNN that combines all the different omics. Here's the architecture of the individual DNN. As you can see from the architecture, there are embedding layers. And why do we need them? One major challenge that we faced was the n smaller than p issue, which is an inherent issue in many genetic data. The data has extremely high dimensionality, but is very low in sample size. And furthermore, there exists complex interactions and correlations between the predictors. And the solution to that is graph embedding of gene interaction network. And there are two benefits to this. First, it can reduce the dimensionality of data, and secondly, genes that are functionally associated tend to be statistically dependent and work in a synergetic fashion. And by integrating the interaction and correlation between the genes into the deep learning models, we can represent the underlying fundamental biological mechanisms of cancers. The figure to the left shows a subgraph of the gene interaction network, which is used as the graph embedding layer. The gene interaction network is obtained from existing biological knowledge of the gene interactions, and the graph embedding layer is added between the input layer and the first hidden layer, and the output of the first hidden layer will now be shown in the formula below. However, for some omics, we are unable to create graph embedding layers because their interaction network could not be created due to lack of existing biological knowledge. And the solution to this is to use a dense embedding. Dense embedding is created to reduce the dimensionality of data. Here we want to investigate the effect of embedding. The graph on the left shows the MSE for a model with embedding, and the graph on the right shows the MSE for a model without embedding. The models with embedding are superior than the models without embedding in the following areas. Firstly, in terms of accuracy, the models with embeddings have better result, which means lower MSE than models without embedding. And secondly, exploding gradient. The models with embedding layer, the loss reduces gradually and smoothly with epochs, and the models without embedding layer, the loss is very unstable with sporadic exploding gradients. And thirdly, models with embedding have higher convergence rate compared to models without embedding. And fourthly, training time. Models with embedding have significantly faster training time compared to models without embedding. And therefore, models with embedding have superior performance than models without embedding. Here we have the architecture of the combined DNN models that use all omics. The input of the combined DNN is the output of the individual DNNs. The attention mechanism allows us to non-uniformly weigh the contribution of the input features. It was originally developed for RNNs, and its purpose is to pay different amounts of attention to different hidden states in RNN. And in this case, it allows us to pay different attention or give different weightage to the weightage of importance to the output predicted by different models. And the attention is calculated as shown below. To investigate the effect of attention, we created two models with and without attention. And as you can see, the model with attention mechanism achieves better MSE compared to models without attention. And this proves that attention mechanism indeed helps to improve the accuracy rate. Since the goal of our research is to be able to predict the cellular responses to various drugs so that doctors can prescribe the most effective drug to the patients, another metric that we have devised for judging the DNA result is the percentage of times that the predicted most effective drug was in fact the most effective drug, and the percentage of time where the predicted most effective drug was among the top 3 most safe drugs in actuality. The most effective drug is the drug with the lowest IC50 value, and the result is shown in the table on the left. The chance of randomly choosing the most effective drug out of 22 drugs is about 5%, and our DNA model is able to increase that chance to more than 50%. In the future, the research can be extended to many other areas. One area that we could look into is feature importance, as currently our models are a black box, which makes it almost impossible to understand the underlying biological mechanism between cellular genetics and drug responses. We could look into feature importance to try and find the correlation between specific features, such as the mutation of specific genes and the effectiveness of drugs. This might allow the researchers to uncover new biological and pharmacogenetic information previously unknown. And another area that is worth exploring in the future is the chemical composition of drugs. We can incorporate chemical composition of drugs as input of our prediction, and we might be able to discover new correlation between specific chemical makeup and cellular biomechanisms. And that is the end of my presentation. Thank you very much.